right, so thanks for having me. It's a pleasure to be back. I've presented a few times here, and in fact, one of the first slides is from a previous presentation because it motivated this work. So first, the talk is in two parts. First, I'll motivate variation graphs. This is a kind of way of representing a pan-genomic data structure, and I'll describe how we develop them and so on. And in the second part, I'll talk about applications to show why it's useful. So over time, genomes copy, sometimes with error. And as these errors proliferate, they give rise to genomic diversity. So we can represent this in the phylogeny or generations. And of course, this is like the object we really want to think about when we think about collections of genomes and populations. But for expedience in contemporary practice, we usually pick one representative copy, which we call a reference. And in this copy, of course, there's only one version of every locus. We don't have these variations anymore. And so the standard practice is to take a reference genome, which perhaps we reference it's just a big string, right? We align new reads to it, and then we can do other stuff. We can do stuff with the read alignments. We could call variants and get genotypes from them. We might be interested in coverage or allele-specific expression analysis. And this is good, but what's problematic, and it's kind of a quiet lie we tell ourselves, is that this is not a real genome because we can't take the variation information, even from a single diploid, and update that object. And so we're always limited in our representation of genomes if we represent them in this kind of simplistic way, although expedient way. And so this is the slide that I presented 5 years ago here. It's from the 1,000 Genomes project. There's a locus, which we found, where there's no real evidence for variation in the primary read data. But if you take a variety of different variant colors, which can build assemblies, you can see that there are three particular alleles at the locus. There is the reference allele, of course, which is matching all these reads. Then we have a large deletion. And also overlapping this is a small deletion of a single base pair. And if we take the reads in this locus that map nearby and maybe don't map well, we realign all of them to this graph and then project them back into the reference, we can just plainly see these alleles represented in the reads. And so if we had this in our reference object, it would be much simpler later to then do variant calling and analysis on this kind of allelic variation. And so one solution to this problem is to build a pangenomic object that lets us consider all the genomes that we're interested in. So we can take the whole phylogeny that we're curious, we can pick all the leaves off of it, and we can take the alleles in them and all the sequences and compress them into a graph. And in this graph, we have, of course, all the genomes we're interested in, and also all the sequence we're interested in. And yeah, so this is a variation graph. And the point of a variation graph is that we don't just have a graphical object describing allowable sequences, but we also remember the underlying haplotypes that we built them from. So here, in this model, we attach sequence labels. So the sequences are on the nodes, and they have excuse me, they have identifiers, and the edges represent allowed linkages between them. So you can kind of simulate genomes by walking along this Markovian model. And of course, because it's Markovian, you lose information very quickly, and so you can hallucinate things that don't exist. That's why we find it very important to maintain information about the original references we built it from. And that's the core concept of a variation graph. It's not just a sequence graph. The haplotypes are important as well. There's many ways to visualize this and think about it. They could be helpful if you're considering using it. Another way is a really path-specific layout mode, so we can think about these bundles of paths. They represent different genomes, and if we walk along a path of a certain color and read off the node labels, we'll reconstruct that genome. And then there's another perspective which is very helpful in some analytical context, and it's kind of a monoallelic model. So every single base in the reference system is represented as a vector. And we can think about annotations or genomes against this as a kind of one-hot vectors or vectors of continuous values across the space. So in this case, the blue genome here would have this T in this SNP, and then it would contain this sequence that's part of an indel that's present or absent in different individuals. So this is not a new idea. I'm not claiming that in any sense. You could go back to the beginning of bioinformatics and see many methods that utilize this kind of concept. Multiple sequence alignment, assembly graphs, these kinds of things all tackle this sort of issue. But what's new is our idea that we want to scale this up and utilize it as a core in analysis at whole genome scale, and for humans, and also for population scale. So there's a lot of hype behind this. Let's see, for the past, this is 3 years ago. So 3 years ago, they started to appear these articles in the popular science press describing this effort. And so we're going to reboot the human genome because, of course, the first time we made it, it only had one copy, and we know that's a lie, right? So we'll make it have all the variation we're interested in. There's been other stuff on it as well, and it's continued. And in response, members of the community interested in this kind of thing got together, and we wrote a review paper on this topic, and termed the new field that's being reborn computational pan-genomics. And so the basic idea is that you have these objects that represent many genomes. There's many ways to do it, graphs being only one. Of course, that's what I'll be presenting today. And then in response to this, they presented a schematic of all the operations you need in order to do pan-genomics in this new sense. We, in the past 4 years, have developed a toolkit that implements all of these operations. So you need to get data into this kind of system, so we can construct these genomes we can construct these graphical objects from existing data, like VCF files or multiple sequence alignments, assembly graphs. Then, in order to operate on them at scale, it's not enough just to keep them in textual format. We need efficient indices that let us look things up, so sequences and haplotypes. And then you can leverage these indices to map reads or new sequences into them. And that's, of course, the basis of resequencing analysis, a way to do out-of-core analysis in large data sets. And then using those alignments, we can do variant calling. We can then take the variants and actually update the reference system. And we want to interrogate the database that's associated with it. We can visualize it as you've seen. We can simulate reads. And of course, importantly, we can annotate the reference system with existing annotations in other reference objects that we already have. Okay, so the remainder of my talk is about applications of this system. And of course, there's many. In fact, there's some in the abstract that I wasn't able to present, but I'll provide what I can. So we went immediately for the biggest possible problem we could find, which is making an assembly graph, making a pan-genome graph out of a human data set, in this case, the 1,000 genomes. So we have 85 million variants from 25 populations around the world, and we turn it into a graph. And then we do all these operations we want to do on it. We index it, map reads to it, and we need to evaluate what we do. So as a baseline for indexing and construction, we construct the pan-genome, or rather the pan-genome, the linear graph made from the reference genome that the 1,000 genomes was aligned to. There are no variants in it. It takes about an hour. It makes a file that's about 2 gigabytes large. And then we index it for search based on techniques developed by Jouni Sirén, who's really been instrumental in this work, and this generalization of the FM index used by BWA-MEM that works on graphs. And then we can align 10 million reads, and for reference, it takes about 7 minutes to do the same alignment with BWA-MEM, so we're running 5 times slower. We have a more complicated algorithm. And then if we add all the variation in, things get worse, but they don't get exponentially worse. We're still able to do this computation. It's still tractable. It takes us maybe 4 hours to build the graph. It takes us 2 days to do the index. And of course, once built, we can reuse it many times. And the alignment doesn't slow down substantially, although it is definitely slower. And as a point of reference, because we don't need to put all the variation in to make this useful, we established a series of filtered data sets. So we remove allele frequencies below 0.1 of 1%, 1%, and 10%, producing a series of indices. And you can see that as the number of variants get in this graph goes down, the numbers start to look much more like they do for the linear reference. So we can actually build these graphs. And then we want to evaluate how well they perform. So to do this, we can't take a real read data set and know where the read should map, so we have to simulate. We used the genome in a bottle truth set for a sample that was not in the graph. Here I'm showing ROC curves. This is the best possible outcome, lowest false positive rate, highest true positive rate. And there's 2 sets of things we see, single-ended mapping and paired-end mapping. The green and blue are against the linear reference. This is BWA, this is VG, and this is against the pan-genome in red. And you see that you actually don't improve performance using this pan-genome in the aggregate. And it's a little frustrating, but it's important to understand why. And it's because the model is maybe overspecified. It has too much information in it. And we can see that because if we look at the reads that should match the reference, we do worse. So we're actually getting confused when adding all this variation in. But thankfully, if you're interested in variation, which many of us are, you always do better using the pan-genome, even including all the variation at the variant site. So these are reads that are touching any variant. They've been simulated from them. And then to explore the effect of the allele frequency threshold, you can see, unfortunately I have to do this rather quickly and still have information, but I'll flip through. We can see frequency 1/10 of 1% threshold, 1%, and a 10% threshold. And you see that we start to approximate the same performance as the linear reference, but we still have the benefit here. And this basically says, we get a benefit from having things in the graph that are in the sample. And it's the common variants that tend to be in the sample. That's most of what's in there. And just to drive this home with real data, we can take a sequencing data set from the same sample, align it back, and look at the heterozygous alleles in it and ask what is the reference bias at those alleles under different alignment conditions. So we can align with BWA here in blue. Sorry, I should explain this more clearly. So this is the indel length frequency distribution, where 0 is a SNP. And this is the fraction of reads that are supporting the alternate allele at these heterozygotes. So you see that we have basically no bias if we align with BWA mem. It's as expected, but as with SNPs. But as we increase in indel length, there is bias. And for alleles that are not in the 1,000 genomes graph, we have the same kind of effect with VG. It's not doing any better in that sense. But for alleles in the graph, we completely remove the reference bias. Like they're seen as if they're part of the reference. So this is the capability of the method is not just to align short reads to a linear kind of graph. We can also align long sequences together. We can align long sequences to that kind of object. So here we take VG, we take its mapper, and progressively align a series of 114 chromosomes, fully assembled yeast chromosomes that were assembled de novo individually, and making this hairball that is 14 megabases in length. So it's about 2 megabases larger than each one of the individual inputs. And this just shows us that we are actually collapsing things correctly, that there's some accessory portion that we're adding, but in general, the aligner is doing the right thing. And then we can take this progressive assembly and align, we remove one of the samples from it, and we align the PacBio data from that sample back to simulate what it would be like to have a new individual against it. And we can also align the data to that graph. We can align it to a single assembly, the reference strain. And for every alignment, so we do 43,000 alignments, for every alignment we have a point. And that point represents the score in the graph versus the score in the linear reference. And you see this large shift here, which represents that this graph object is a better match for the data. And you also see it in the mean identity, which is about 5% higher. Another place where it matters a lot to avoid reference bias is in the case of ancient DNA. So here we have very short reads, very high error rates, and we don't really have much capacity to mitigate that issue. And you can see that this reference bias is quite pernicious, because if you simulate reads from alleles that are in ancient genomes, we actually use a tool called Gargamel, so the read error distribution is as expected. Then map them back. We can do the same kind of allele balance metric. Here, 100% would mean we're aligning 100% of our reads to that particular haplotype. We divide them into reference and alternate haplotype, and we see that for BWA, as the error rate increases, the damage rate increases, our reference bias increases dramatically. And so 20% error, we're missing 10% of the reads that should align to that allele. And this may not matter so much in high coverage, but it certainly is an issue in lower coverage data. Whereas this is not the case for VG. When aligning against the 1,000 genomes graph I presented, there is no distinguishable difference in the amount of reads we're mapping for both conditions. So we completely remove reference bias in the case of this high error data. And we can get some sense of whether this is true in actual data by taking a high coverage individual and mapping reads back to the pan-genome and then calling variants from it. So these are variants we call in the 1,000 genomes, that match variants in the 1,000 genomes, and these we call that are not. And we see that if they're in the graph, we completely remove reference bias. We have a completely normal distribution here. We're observing equal amounts of both alleles. This is a slight change. Actually, it's a bit of like a regression, because the word pangenomics has been associated with microbiology for some time. And the basic theory is that there's a pan-genome that includes all the genes that are interesting in some set of strains, and some are in the core genome. They have to be in all the samples, and then some are in an accessory genome. And there's kind of there's some set that you just have to have for the life and not. And we can get some access to whether or not you can see this with VG by building a pan-genome from Illumina data. We make an assembly with Minia3. This is a De Broglie graph that's been compacted. And then we can align read data back to it and look at the read coverage per sample to make a kind of colored graph. And this is just a tiny fraction of the whole set. I've linearized it, so we made a sort that linearizes the graph. And here we look at 20 KB, and you can see there's this portion here that's shared among most of the samples. And then there's this region here that's shared among 3. And in fact, it's more sparse. There's actually strain-specific components in it. And what this kind of gets to is that although these graphs look complicated topologically at the large scale, we don't have to think about them in downstream analysis that way, because there are often linearizations that make sense, because they are derived from real DNA sequences. And so a very nice thing we can do is to linearize the graph and then think about data against it. And what I'm showing here is that you can basically avoid variant calling, and in fact, just reading through the read coverage for these alleles will show you who is a heterozygote, who is a homozygote, for what SNP. And there's a very full information way to think about the data that avoids all the complicated post-processing steps that we had to do, because we're projecting the data into a uniform basis space that has all the alleles we care about. And so with that, I'd like to thank, in particular, all the people who've worked on this project. It's very much a shared project. And in particular, Jouni, Sirén, without whose work the indexing techniques would not be possible, and my PhD advisor, Richard Durbin, who has been a fantastic advisor, and I can only say good things about. So with that, thank you, and I'll take questions.
Questions for Erik. Start here.
Very nice. Over here on your left.
Hey, thanks.
There's this other effort by Gil McVean and Jerome Kelleher to describe large collections of sequences with ancestral recombination graphs. And I can think of various pros and cons to these alternative descriptions of population samples of sequences. I wonder if you could just say a few words about the trade-offs between a more evolutionarily explicit description of a population sample of sequences and a description like this that's sort of more geared for mapping problems and that sort of thing.
Yeah, I think that they can work together. That's one side. So I don't think that the two approaches have to fight. But there could be advantages if you don't really have a lot of data, or for example, you don't have haplotypes. You might want to use something that just represents a graph topology. But on the other hand, we are really interested in making the graphs represent evolutionary histories. And so probably we should think about trying to integrate with that effort. I don't think that they fight with each other, though.
My question may be a bit naive, but so you told how to look at known variants using this graph, so to map reads on known variants. Does it also help to call novel variants?
So unfortunately, we've always thought that would be the case, and I just have to say we haven't really proved it to be true. And most of that's just down to the experimentation. It took a really long time to get it to work, as expected with just the known variants we had. I guess you could argue that it should be the case, because if the graph is a good model for the individual, then you'll have a smaller delta against the new sequence for the novel variation. But I can't claim that we've actually shown that.
So the more variants you include in the graph, the more likely it will be that you map a read to a non-unique location. So that non-uniqueness will hurt you at some point. So did you look, to which extent does it hurt you, and how does it affect your evaluations? It seemed like you only looked at sensitivity of finding that read and not at that uniqueness property.
If I can go back. Okay. I'll try to go back quickly and show this. But I went through pretty fast. So this is also telling about specificity. These are rock curves. So if that helps, you lose sensitivity and specificity, actually. And this is pretty much true until you're including just variants that you really expect to be in the sample, right? Like at 10% frequency. It's not really an answer to your question, but this is really obviously a problem, and it's been a reason some people haven't wanted to pursue this. And the solution, I think, is to utilize the haplotype information we have more directly in the mapping. And that's actually a development that's ongoing. It's just not something that I can present yet. And it does help. It does make a big difference. So in that case, you mitigate this issue as much as you can.
Okay, I think in the interest of time we should move on. Let's thank Erik again.